Hello, everyone. Welcome back to Meds.Concepts. Today, we will discuss an important molecular biology topic, gene and genome. Genes are the functional units of heredity, while the genome represents the complete genetic material of an organism. This topic is extremely important for CSIR-NET because it connects molecular genetics, evolution, genomics, and virology. In this lecture, we will study the evolution of the gene concept, introns, gene duplication, homologous genes, pseudogenes, genome organization, mitochondrial genomes, viral genomes, and important high-yield concepts. Evolution of gene concept. The concept of the gene evolved gradually with advances in molecular biology. Initially, Beadle and Tatum proposed the one gene one enzyme hypothesis based on experiments in neurospora. Later, it became clear that not all proteins are enzymes, so the idea was modified to one gene one protein. Further research showed that many proteins contain multiple polypeptide chains, leading to the modern one gene one polypeptide concept. Today, gene is defined as a functional DNA sequence capable of producing either a functional RNA or protein product. Importantly, not all genes code for proteins. Some genes produce functional RNAs such as rRNA, tRNA, and miRNA. Split genes and introns. In eukaryotes, many genes are interrupted by non-coding regions called introns. Such genes are known as split or interrupted genes. The coding regions are called exons. During RNA processing, introns are removed and exons are joined together through splicing. Genes without introns are known as uninterrupted genes and are common in prokaryotes. Introns may vary greatly in size, ranging from about 50 nucleotides to several kilobases. Some important genes lacking introns include histone genes, interferon genes, and certain heat shock protein genes. Introns are biologically important because they allow alternative splicing, increasing protein diversity. Gene duplication and evolution. Gene duplication is one of the most important mechanisms driving genome evolution. When a gene duplicates, one copy may continue performing the original function while the other copy accumulates mutations. There are three major outcomes after duplication. First, both copies may retain the same function. Second, one copy may acquire a new related function through divergence. Third, one copy may become non-functional and form a pseudogene. Gene duplication provides raw material for evolution and contributes to genomic complexity. The globin gene family is a classical example of evolution through gene duplication and divergence. Homologous genes. Genes that share a common evolutionary ancestor are called homologous genes. Homology indicates evolutionary relationship rather than simply sequence similarity. There are two important types of homologs. Orthologs are homologous genes present in different species that evolved after speciation. They generally retain similar functions. Paralogs arise due to gene duplication within the same genome and may evolve new functions. The globin gene family, including alpha, beta, gamma, and delta globins, represents paralogous genes generated through duplication events. Pseudogenes. Pseudogenes are non-functional copies of genes derived from functional ancestral genes. There are three major types of pseudogenes. Unitary pseudogenes arise when functional gene loses activity without duplication. Duplicated pseudogenes form after gene duplication followed by mutation accumulation. Processed pseudogenes originate from reverse-transcribed mRNA molecules that reintegrate into the genome. Processed pseudogenes usually lack introns because they are derived from mature mRNA. Most pseudogenes are non-functional, although some may acquire regulatory roles. Gene families. A gene family is a group of genes having similar sequences and related functions. Gene families mainly arise through repeated duplication and divergence events. Examples include the globin family, histone family, and ribosomal RNA gene family. The globin family is divided into alpha and beta globin clusters. The alpha cluster includes alpha, zeta, and theta globins, whereas the beta cluster includes beta, gamma, delta, and epsilon globins. Gene families allow specialization of gene functions while preserving structural similarity. Acquisition of new genes. New genes can arise through several mechanisms. The most common mechanism is gene duplication followed by divergence. Another mechanism is horizontal gene transfer, where genes move between unrelated organisms. This process is especially common in bacteria. A classical example is Agrobacterium tumefaciens, which transfers DNA into plant cells. Genes may also arise de novo from previously non-coding DNA sequences. These mechanisms increase genetic diversity and contribute to evolution and adaptation. What is genome? The genome refers to the complete genetic material of an organism. It includes not only genes but also regulatory sequences, repetitive DNA, and non-coding regions. Cellular organisms generally possess DNA genomes, whereas some viruses contain RNA genomes. Thus, the genome represents the total hereditary information required for structure, function, and reproduction. Importantly, the genome is much larger than simply the collection of genes because non-coding DNA occupies a major fraction. Prokaryotic and eukaryotic genomes differ significantly in structure and organization. Prokaryotes usually possess a single circular double-stranded DNA chromosome. Their genomes are compact and contain very little non-coding DNA. Many bacteria also contain plasmids, which are small extrachromosomal DNA molecules. Eukaryotic genomes consist of multiple linear chromosomes associated with histone proteins. Eukaryotes contain large amounts of non-coding DNA and introns are very common. Bacterial chromosomes are negatively supercoiled, which helps compact DNA efficiently. The Human Genome Project was an international scientific effort aimed at sequencing the entire human genome. It started in 1990 and was completed in 2003. The project revealed that the human genome contains approximately 3.2 billion base pairs and around 20,000 to 21,000 genes. One surprising finding was that only a small fraction of the genome actually codes for proteins. Large portions of the genome consist of repetitive and non-coding DNA. The Human Genome Project revolutionized genomics, personalized medicine, and disease research. The human mitochondrial genome is a small circular double-stranded DNA molecule. It is approximately 16.5 kilobases in size and is maternally inherited. The mitochondrial genome contains 37 genes, including 13 protein-coding genes, 22 tRNA genes, and 2 rRNA genes. These genes mainly encode proteins involved in oxidative phosphorylation and ATP production. Unlike nuclear genes, mitochondrial genes generally lack introns. Because mitochondria are inherited maternally, mitochondrial DNA is widely used in evolutionary and forensic studies. Viral and viroid genomes. Viruses possess highly diverse genome structures. Their genetic material may be DNA or RNA, single-stranded or double-stranded, and linear or circular. RNA viruses may have positive sense, negative sense, or ambisense genomes. Positive sense RNA can directly function as mRNA, whereas negative sense RNA must first be transcribed into complementary RNA. Viroids are even simpler infectious agents composed of small circular single-stranded RNA molecules. They do not code for proteins and mainly infect plants. Retroviruses such as HIV possess positive sense RNA genomes along with reverse transcriptase. Genome organization in viruses. Viral genomes may be organized in different ways. Monopartite genomes consist of a single nucleic acid molecule. Multipartite genomes are divided into separate nucleic acid molecules packaged individually. Segmented genomes contain multiple genome segments within the same viral particle. Influenza virus is an important example of a segmented RNA virus. Segmented genomes are biologically important because they allow reassortment, which contributes to antigenic shift and emergence of new viral strains. Finally, let us revise some important high-yield concepts. Split genes contain introns, while processed pseudogenes lack introns. Orthologs are homologous genes present in different species, whereas paralogs arise through gene duplication. Gene families evolve mainly through duplication and divergence. The human genome contains approximately 3.2 billion base pairs. Human mitochondrial DNA is maternally inherited and lacks introns. Retroviruses poses reverse transcriptase. Influenza virus contains a segmented RNA genome. These are frequently asked concepts in competitive examinations. Thank you.